Thank you very much, ladies and gentlemen. We're going to introduce ourselves first and then jump right into the discussion. I am a journalist, editor of MedNous, which is a business science publication, co-founder of this publication. Before I did this, I was editor of 2 regulatory publications. I thought I'd throw that out because regulation doesn't often come up in these discussions. And then prior to that, I was a business and science correspondent in Amsterdam, Brussels, and London for 2 international news organizations.
Great. Also, my great pleasure to welcome you to this panel discussions where we guide you through some of the perspective of the future of gene editing. And really, just briefly about myself, my name is Tim Lohoff, Senior Associate at Forbion, and been involved through our portfolio work in this field quite intensely, and really wanted to pay the attention to our panelists and shout out a big thank you for joining us today, and would like to introduce them from left to right, starting with Albert Seymour.
Great. Thanks, Tim. Thanks, Victoria, for the opportunity to be on the panel and for the organizers of the event. And so my name is Albert Seymour. I'm President and CEO of Seamless Therapeutics. We're a company that's working on foundational technology to program what are called recombinases. And so these are a new set of editing enzymes that allow us to edit the genome in very specific regions of the genome. And as the name implies, they're very precise and within the edit. So it's right to the nucleotide. So it's seamless, if you will, when we go into the edit. I've been in the industry for almost 30 years. And so it's we've seen some ups and downs and things like that. And it always sort of rebounds. And so I'm looking forward to participating on the panel and talking about the future of gene editing as well. So.
And my name is Avencia Sanchez-Mejias. Thank you so much also for the invitation to this panel. I'm the CEO and co-founder of Integra Therapeutics, that is a preclinical stage and Barcelona-based biotech. And we do technology development for programmable transposases in a similar manner. In this case, we combined the gene editing technology of CRISPR-Cas nucleases with DNA transposases for doing programmable and precise integration or gene writing. And what we can bring into the space is the ability to handle very large DNA cargos that can be large causal genes or multicistronic therapeutic cargos. And we can deploy these both for cell engineering, ex vivo and in vivo, and also for gene therapy. I'm very happy to be here.
Okay, so hello to everyone. I'm Monika Paule. I'm the CEO and co-founder of gene editing company Caszyme. And we are CRISPR experts, as we call ourselves, working in this field from the very beginning. And what we do, so we develop novel genome editors and have a platform of these unique tools that can be applied first of all in the therapeutics, but also even in the other fields like diagnostics, agritech, and so on. And we collaborate with companies as Integra, for example, in applying our tools to develop gene therapies or cell therapies and other and products.
Hi everyone, my name is Paul Vogel. I'm Vice President of Research at AIRNA. I'm also Managing Director of its German subsidiary. We are a transatlantic company with sites in Cambridge, Massachusetts and Tübingen, Germany. We are an RNA editing company using chemically modified oligonucleotides that can direct the endogenous enzyme ADA for making very specific changes, genetic changes at the RNA level instead of the DNA level, which comes with certain advantages. I work on this technology for more than 10 years, started my PhD at Thorsten Staffel's lab, the pioneer of RNA editing, where we saw the whole evolution, which means we translated this first from reaction tube to cell culture. I later went on to Stanford University to do my postdoc there with Jin-Billy Lee, which is another RNA editing expert, where we translated this technology into a mouse model of alpha-1 antitrypsin deficiency, which is our lead program. And then to 2022, I co-founded AIRNA together with Torsten and Billy and my colleague Tobias. And now I'm focusing my work on finding new targets that which uniquely suitable for RNA editing.
Hi, I'm Sean Kendall, chemical engineer by training, worked in the synbio space for a while. Been at Arch Venture Partners since 2016. I moved out to Europe in just about a year and a half ago to expand our efforts out here. We were investors in AIRNA here and longtime believers in the gene editing and gene therapy spaces broadly. Excited to be here.
Right, okay. So I thought I'd start off by letting you know that Tim and I have only just met as a consequence of this panel discussion. And what we discovered about each other is that as an employee of Forbion was, Unicure was a portfolio company of Forbion when Unicure developed Glybera, which was the first gene therapy, which was a gene replacement therapy. And it was approved and put onto the market in 2017. Now, I was writing about Unicure at the same time, so we had a delightful conversation about what's happened between gene replacement that got onto the market and then had to leave, and what's happened now. And so it's slightly ironic that only a week ago, Bluebird Bio had a similar experience, much different product, much more sophisticated, 3 products on the market, but it did have to withdraw basically from the market, go private basically. And so our first question, which is going to be directed at Albert, is how have things changed during that long period, well, not so long period, between the first replacement therapy and today?
Yeah, I think it's a really good question. It's something that it's nice to be able to look back on the history of that as it's coming along, because even the history even before Glybera was onto the market, which is so much, investment and research into gene therapy. And so what you saw with Glybera was that it was sort of the first approved product utilizing an AAV, which was a new, at the time, way to deliver gene therapy into cells, particularly non-dividing cells, so you could express that. And they went after a very rare disorder where they could express an enzyme called, in this case, it was LPL, so that they could sort of reduce fat in the blood. And at the time, it seemed like that's a great sort of progression for that particular arena. I think they ran into some issues with the number of injections that they had to do, and so the administration of it, the manufacturing of it, was very expensive. The amount of R&D that was required in order to get there was also very expensive. So then on that, once you go to commercialize it, the cost of goods were high. I say that because now, as you move forward several years, that's actually starting to come down. And I almost look at gene therapy and these delivery approaches in the manufacturing similar to antibodies. So antibodies started off in a similar situation because it was being compared to small molecule type therapies, which was very inexpensive to make. But antibodies at the time, as a first biologic, were very expensive to make. And then you saw over time that people realized the advantages of a biologic with respect to the antibody. That cost of manufacturing was reduced significantly. So then you now tie that into some of the other approaches utilizing gene therapy. You're seeing some of the manufacturing come down. You're seeing new companies now really focus on what is the technology that you have, what's the right fit for the disease that you're going to go after, and as you move into development, to make sure that you can sort of be maybe fiscally responsible with respect to how much is it going to cost to get through development, representative of the indication that you're going to treat. And I think those are some of the key learnings that you come out of that, because coming off Glybera, you also saw that it can work. It did work. Clinically, it worked. And then you move in, and the next one I think that was approved was Luxturna. And there was another one that was a direct injection into the eye. And when you did that, it was a very rare population, but the biology was very well characterized from a genetic perspective, and it really worked. But again, they also face it's an extremely rare indication. And then coming off that, you have Zolgensma, which now I think is more of a success case because, again, you knew exactly what the gene was. It was a huge unmet need, and you were able to deliver this into babies and pretty much allow them to live much longer.
And I
so
think
you progressed along that.
Zolgensma has a new indication as well, recently.
Yeah.
Yeah.
Yeah. So anyway, that's, I think, from some of those learnings. So as we sit here with the new technologies that we're facing today, I think a lot of thought goes into what does the technology offer that's completely differentiated from something else? What's the unmet need that you're trying to address? And can you get there in a cost-effective way in order to deliver it? And so if you think about all the way back to Glybera and then all the learnings there, I think that's what a lot in the community are now embracing and learning from some of those earlier learnings.
Thanks, Albert. So if we zoom out a bit and kind of project on what we have learned, as you pointed to, for the gene editing field, I think we clearly can look back on an evolution of a toolbox that has been established over the last 10, 15 years, where we have already seen some of the same mistakes to an extent being applied. So I think I would like to ask Avencia to give us a bit more on her perspective on how this toolbox really has come together and how we can now take these learnings of indication selection into consideration when we think about the gene editing as a modality class.
Yeah, I love this question because being in this field over the last 4 or 5 years has been very fun to see how the toolbox has increased and see we are talking about the last 10, 12 years, but there was some more old technology for doing gene editing before CRISPR. But the beauty of CRISPR is that without doing any protein engineering, you can very fast change the target, and this has made more accessible the gene editing field. And it's also funny how we call CRISPR gene editors, but actually they don't edit anything, they just cut. And it's the actual cell that does the edit. So I think we have to keep this in view, because depending on the cell, this edit will have a different outcome. And this is important. So CRISPR, the first generation of gene editing tools, does cut and destroy for the most part. And they are very good at doing this. And we are starting to see the first clinical assets being approved based on cut and destroy, that sometimes you don't want a gene to be expressed. And when we build the pipelines, we have to think on the potential of these toolbox and how different they are. And then comes the second generation of gene editing tools, in which you actually add an editor to these scissors. And we have prime editing, base editing, to change maybe one nucleotide, not all possible nucleotides, but some of them. Then with prime editing, some more capacity, a few letters. You can also add epigenome editors that here in this panel we don't have representation of epigenome editing, but this is also a very nice way to modify the expression of the genes. You silence, you activate. And now in the last 3 to 4 years, we have the expansion of the toolbox with the gene writing tools, the third generation, in which we can add transposases, like in the case of Integra to this editing, and you can have a standalone editing tool such as recombinases that you can engineer these proteins to be more flexible. Also the editing at the RNA level. And each tool will be better suited to fix one specific problem. And sadly, there is a huge unmet medical need in especially in rare diseases, but in other indications as well that we will touch on during this panel. And there is a space for all these tools, and you very well pointed at it, that we have to choose the right tool for the proper problem. And be very mindful on how we use this, because not all tools will be able to do it all, but all together as the toolbox expands, we are in a very good position to now target additional diseases.
Thank you. And Albert, you brought up an important point, which is the learnings from delivery by using viral vectors, which is certainly an important component when we speak about gene editing. It's kind of bottlenecks on really bringing it to a product level. So this is a question to the entire round. Would love to hear your view on how we can combine the power of this really immense toolbox you have just outlined to really find the right delivery approach to get to the products and applications we all seek treatments for patients for. maybe Sean, you can share your perspective.
Sure. Yeah, I think one of the things we've seen in this field that I think we tend to see in most new modalities is a little bit of the kind of 2 steps forward, 1 step back kind of world where, you know, at first just the sheer capability of a new platform is remarkable, and the things that you think you can do with it before you kind of run into some of the new either new biology or manufacturing or delivery questions. Which you don't always, you know, sometimes you bet on the world to solve some of those for you, and sometimes those nuts are harder to crack than you think. I think is a phenomenon we tend to see in the space, and as part of the evolution. I think Albert made the point earlier on that you know, sometimes these things eventually get ironed out bit by bit. I think one of the things that we've seen recently is, I mean, AIRNA and Paul, I'm sure will cover it at a deeper level. I think part of the excitement there was really around opening up, being able to do temporary editing, because you're drugging just the RNA. You're making a therapeutic that's eventually going to be eliminated from the system. But you know, as we've seen other challenges come up kind of broadly across other modalities, some of the other advantages in terms of manufacturing and delivery that are solvable now for that particular modality, and I think will be coming up for many others in the future, I think has been actually something we've found increasingly advantageous.
I just want to step in for a minute and mention the lentiviral vector, which a lot of people were criticizing a few years ago for various reasons. I was very interested to learn about Novartis's work with a U.S. company called when they're investigating a lentiviral vector that could target and transduce resting T cells, which would then become products. This is a company, I'm now speaking about Novartis, which pioneered the CAR T. And now they're going back and they're saying, hold on, there's something more that we can do for the CAR T, and that is to change the vector approach. And it's basically an in vivo approach that they're looking at now.
Sean, you also mentioned like the learnings of other adjacent fields like RNA interference by using GalNAc conjugated RNA to really, first of all, have a transient product, but also to an extent take the learnings of delivery there. So Paul, maybe you can share your real life experience with this modality class and how it has enabled you to really kind of develop transformative therapies at AIRNA.
Yeah, I think so. Our advantage is that we are using chemically modified oligonucleotides, and so we can really take the advantage to learn from the advances made from siRNA or ASOs that has been developed for the last 25 years, which means we can use basically the same chemical modification to make our ASOs more stable, but also basically using the same delivery vehicles. And one of those promising vehicles are GalNAc. So we normally conjugate our ASO with GalNAc so that we basically get a very specific liver targeting and liver delivery. And this also enables us that we have a very long mechanism of action compared to LNPs, for example. With LNPs, you normally have to dose once or twice a month. With GalNAc, actually, you can basically dose a quarter, or from a siRNA perspective, sometimes you can also dose twice a year or once a year. So this helps really for us that we have a very de-risked modalities with oligonucleotides. The mechanism is new, but the things we are using has been already developed, and we can also look into the field, and there are new delivery vehicles developed, like transferrin receptor targeting molecules, but also lipid chains like C16 that you can also go into the muscle and also into CNS. So I think in that regard, it is really helpful to work with oligonucleotides, as in our case.
And you mentioned in your daily life, one of your tasks at AIRNA is also to screen for new applications for the technology. I think we have early on in the discussion touched on the commercial consideration for a successful product or to be a viable product in that space. So what is like your way of thinking on really pairing a powerful technology then with a indication or disease that you want to treat to ultimately make an impact?
Mm-hmm. So when we look at diseases, we have first to think about what kind of tissue we can reach. And as I said, we mostly use GalNAc because this is the most de-risked delivery, and this is then basically liver disease. And then we have to see where are the kind of unmet needs. And we do not really want to compete with siRNA or something like that. We really want to find unique indications for RNA editing. And one of those is alpha-1 antitrypsin deficiency. This is our lead program. This is 90% of the cases. This is caused by a G to A point mutation. And this is a kind of gain of function and loss of function at the same time, which means with a gain of function, this mutation makes the protein prone to form toxic polymers in the liver, so patients can develop liver cirrhosis. But at the same time, it's also a loss of function because of this polymerization, it cannot reach the lungs anymore, which to protect those lungs from proteolytic damage. So people also can develop, and this is predominantly the lung disease, COPD. And there are certain modalities like siRNA, which tries to reduce the polymers in the liver, but that's only help the liver phenotype. And there are also certain modalities, clinically advanced, like gene therapy or enzyme replacement therapy, which tries to tackle the lung disease. But there's no modality which can actually do both, address both. The lung and the liver phenotype. And with our modality, we can basically address both by repairing this G to A point mutation back so that we can address the liver and the lung phenotype, basically.
I just want to bring the conversation back to CRISPR-Cas9 because this is the beginning, as it were, of gene editing, and ask Monika to explain to the audience what her company is doing, which is creating new nucleases and also getting patents for them and investigating the size of the nucleases, what they can do, and then at the same time mention your collaboration, your licensing agreement with Integra. It would be very helpful.
Yeah. So we probably are, those as some people who work in the gene editing field for many years called us the old gangsters of CRISPR because we were there before CRISPR was anything. And actually, it's a very interesting story to see from a perspective because, it started, about 25 years ago. But at that time, CRISPR was discovered, Cas proteins also, but what do we do with them? We don't know to full extent. Not the rest of the science hasn't moved that way. And when my co-founders, the scientists in the field, picked up this topic about 15 years ago, it was also very much driven, by the scientific interest. Okay, so how actually all of that works? And 15 years later, we are discussing now, as Avencia put it, first generation, in 15 years. However, when we look at the same time what companies are using, it's very often that original Cas9, which has a lot of, advantages, but many disadvantages as well. And the whole industry was moving, we don't know anything. Now we characterized Cas9. Now, all the problems are solved. Well, they're not solved. We are still sitting here and discussing one or another approach is better. But from our perspective, as being in this field for so many years, it's, all the approaches are great because we are targeting many, many different indications in our complex body. And, we even cannot tell that from delivery perspective, one or another way is way better or way worse. We should talk about combination. So that's where we as a company come in, because we help to develop those tools that are the most suitable for each and every application. And let's say in case of therapeutics, what we are solving is, yes, the size of the nucleases, their cutting capabilities, and how they are, how well they work in combination with one or another delivery technology, and all those bits and pieces that might go wrong on the way until the clinical stage. Because we have the saying that, devil is in the details. So if you don't work on these details, then you might fail. And I think, as you have mentioned at the very beginning, all these failures, it was, first of all, a lot of money poured in the field with just go, 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 and give us, a huge payback. And well, it's still a very scientific field. It's not, producing or making a shoe, which still might not work because it might be, not suitable for you. And on the way, we lose all these aspects that eventually lead that maybe we don't have such a good results. And in general, this understanding where we are going. So as you mentioned that, we have this partnership with Integra. And the way we work is, we work step by step, understanding, each other businesses, scientific, technical capabilities, and finding the best fit. So we are a partner for Integra with our Cas12L nucleases platform that Integra is using when building your therapeutics pipeline. And this, collaboration and understanding of every bits and pieces on the way, I believe will make Integra a very successful company. So that's what CRISPR experts do.
Thanks for sharing that view. And yeah, I think Avencia, you just unmuted. It would be great to hear your view of the partnership.
Yeah, not only on the partnership, that for us, it has been very successful because we started with the science and the validation, and we have a great tool that outperforms SPCAS9 with FICAT. And most importantly, that we can get commercial access in a platform manner, because we are a technological platform. We can do many different things with proper funding, but we can do many things, and we don't want to go asset by asset licensing to the first generation. And I just wanted to comment that this has been a big failure for the patients, having the first generation of SPCAS9 being so blocked. And it's not even helping the businesses that own this technology, because it's not blocking others from building the same solutions. And it's making the final product much more expensive for many. So I think this, getting back to the beginning of your intervention, Albert, it's very important that we don't have to reinvent the wheel every time. And through these collaborations, but also others for delivery. And we have very unique technology that is just a piece in the puzzle of the final product. And it's not sustainable for the industry for us to, and each and every one of us have proprietary LNP platforms and companies. So these have to be rethink, and this also challenge the VC business model. But we have to make it work for the patient and the system, and not to launch something that is not being financial, making the numbers that we expect it to have.
So could we address what is the current market sentiment in your experience? We mentioned the Bloomberg Bio episode, which may be not followed. It may not. It may be. But should we be paying attention? What should we be paying attention to in order to make these products more commercially successful?
To me, I think one is the cost of the R&D and also the cost of goods. But the great news is that the COVID vaccine was viable, and there is not that much different in the product design of a gene editing therapeutic product than COVID vaccine. So with the proper scale, it is feasible to lower the cost and then to make sure that there is a market there. And this morning we heard of the regulatory path for rare diseases being easier for first-in-class modalities, but there has to be a market there, if not for the first product, at least for the pipeline or the first few products of the pipeline. And also think about the scalability and the IP perspective. And this, we will see how artificial intelligence and design of proteins change the patent protection, because sequence of protein is no longer relevant, I will say. And we will see how we adapt to this new reality. But I think it's a combination of both things, but happy to hear about others.
Maybe just one comment, and I think it's just historical when you look at it as well. A lot of these new technologies that were being developed were really focusing on the right indication. A lot of times that right indication was a very rare disease. And so I'll use Luxturna as an example. That one is a very rare eye disease. And there was this thought early on that let's take and focus on that as for lack of a better word, for a lost leader, or get into the clinic, show your technology works, and then go back and do the other. The challenge that we ran into, though, with that is you would invest a lot into those. You would show that it works, but then in order to come back, there was really no commercial opportunity for those indications. And I think that's what a lot of companies, the bar's gotten a little bit higher. The technologies are better, more sophisticated, but the bar's higher on that front, that we need to be more creative on how can we get into indications that not necessarily just to show that it works, but to take it all the way through commercialization, whether you do it by yourself or through partnerships. Because I also think a lot of companies now are much more open to how can we partner in order to get, with the example that you and Monika just brought up as well.
Yeah, I'll echo that thinking. I mean, I think these days what you're saying is a lot of like make sure that first indication works well and lets you earn the rest of the platform, and you're being judged on that a bit more than before. I think and that means it is important to focus back from the disease to kind of find that fit as best you can. I think the point also in terms of what I'm sure everyone here is familiar with is you get to finance, whether it's grants or investment money, what you get to build, what you can raise for, and so I think looking to what those kind of unlocking moments might be that can kind of help you get into other new areas. I mean, I think COVID vaccine's a prime example of something where some of those technologies one thinks of delivery, and there was a time when if you were working on LNP or delivery, people would have just been like, how are you going to make any money on that? And I think that, keeping an eye out for those moments that can be unlocking and seeing if you can kind of tie yourself to that is I think a helpful thing to do.
Yeah, and Sean, picking up on the unlocking event, obviously Caszyme was one of the first gene editing products approved recently. Do you think this is one of these unlocking events, or is it still to be proven based on commercial success of the product? How would you see that?
That's a good question. I think we are generally speaking, we're in a bumpy time in the world market and so on. I think that I think a lot does remain to be seen personally. But I think it's not. Unfortunately, I think it's also some things are within the control of the people developing, and then others are kind of markets broadly. I think it's kind of the responsibility, I think, of all of us to be kind of clear about what can be achieved and what one should expect, and try to deliver as best we can on it.
Great. I think we have heard also throughout the panel how the importance of partnership and living in this bumpy time, as you have pointed out, certainly has some operational constraints that I'm sure some or almost all of you facing on a day-to-day life. Can you maybe share a bit your experience, how we can also grow in this period, really like taking a step back, but finding the right solution that ultimately accelerate the field as it matures? And we obviously want to look at this panel also in the future. I think someone want to go first? Otherwise,
I do. I want to bring the Novartis case back. I really like that. I mean, here's a company that was very successful with CAR T and is now going back to work on it. And who knows, it might be a success. I mean, they've certainly got the commercial infrastructure to deal with it. Sorry.
Well, I will go first. It's a tough question. So I think we have to focus on what our technology can do best, and then create value around that with the resources that we have that sometimes are not as much as we would like. And in our case, going back to the lentiviral platform, we also leverage academic collaborations to get access to new technology that can help us to get this FICA tool into the cells of the patient instead of having to do the whole engineering ex vivo. So leverage not only biotech collaboration, but also academic collaborations within Europe, that there is a public funding for these projects is also a good way to leverage and thrive in these last years, which has not been easy, and really focus on what the technology can do best, because sometimes one clinical trial will fail unrelated to your technology and another one will succeed. So just keep pushing the development and believing on the value that you're creating, and I think that's the way Integra is doing.
I think when we pre-discussed this panel, I think one commonality that we have seen across is also the transatlantic spirit of the companies present here. How do you think like the diversity of like operational skill sets and expertise in this field will help you to really come out stronger? Maybe a question for you, Albert.
Happy to start on that one, because I think and so Seamless as a company, we do have a site in Dresden, Germany, and then also right outside Boston. And the way that it was set up is in Dresden, that's where the technology came from, out of Dresden University. A guy named Frank Buchholz in his lab, and then Felix Lansing was a graduate student in there that really developed it. And so by taking advantage of all those experts right there in incubator lab space, we can have access to not only the current technology that was there, but also new improvements on that technology from the group that does it. And so you can build that out in the German infrastructure out of Dresden. The other thing that can help along those lines is taking advantage of some of the public funding that's available. So being in the US, but then also being in Europe, we can get access to public funds to help build and foster biotech in the German space. And so we've been able to utilize that as well. And so it really helps us keep that expertise close to where it was originally discovered. And then in the US, build out what's really, I think, unique, and we can take advantage from the US perspective, and that is maybe access to CROs, animal models, things that we can do from a preclinical perspective, regulatory and clinical, that there's a whole big infrastructure that built ready to go in the Boston ecosystem. And so it really helps us take advantage of both sides of the pond. The last thing I'll say is, as we get closer to the clinic too, when we think about moving into clinical trials, there are a lot of regulatory agencies that you can think about working with. And so you can be in the US, you can be in Europe. Having a facility in both of them really allows you to do that, because when you go and talk to regulatory agencies and you move, you want to have some person on the ground in at least Europe versus US if you were going to go for that. So it's allowed us as a company to think not only how can we take advantage of it now, but also in the future as we go.
Maybe I can also say something about that. I have actually the same experience like Albert has. So our technology comes from Tübingen University, and people like me or Toby who worked on this technology joined the company there, and this really helps to move science forward for the company. But on the other hand, having a US site also helps in terms of getting managing experience, people who have done this several times, and help you also to grow the company. You get also a different mindset. It's not only that you have always the conservative German mindsets in the company, but also this very great American driving experience. So I think if I would have to do it, I have to found a company again, then I would do it with the same way we did.
I might just won't say a whole lot that hasn't already been said, but I'll just kind of echo. And it's neat in the way that, I mean, ultimately any of these things comes down to a people business. And I think what we've seen from just the company gets built based on where we're able to get the people. And I think the talent pool in Europe is super deep. The talent pool in the States is deep. If you can take advantage of all of that and you do have to manage, of course, working in different time zones and these things that do add a little bit of complication. But if you're really able to build a world-class team, and by going around the world to do so, I think that's we found that highly advantageous.
Just on the question of pricing, which is where this came from, I'm wondering if we could have a little conversation about the once-in-a-lifetime treatment and how that expectation is priced currently. Because gene and cell therapies are unique products in the sense that you're being asked to pay up front for a hypothetical outcome, at least until we get another generation of patients where we can actually have some real-world data about what the duration of treatment was. And I think that's one of the issues that's kind of clogging up the industry, because people have this wish or this expectation that it's going to be there for lifetime, and then they pay a lot of money, and I don't know how you can sell anything on that basis, but your views would be appreciated.
Maybe I can jump in with that. And I think that's also very much related, the price level with these ups and downs in this field, because the big bottleneck that we see now after all this money invested into the field, that the actual outcome is extremely expensive. And when you compare that, yes, let's say with CRISPR and gene editing and all these therapies that people around this table are developing, you can expect that it would be like a one-time treatment, which means you are cured, which you think, okay, that, you can put a big price tag on that. But at the same time, what we know, talking with companies, with hospitals, with patients, there is some reluctance for this one-time treatment, because, as you said, you don't know what the effect will be. And especially when there is an opportunity to use some kind of a treatment which is continuous, but let's say still allows the patient to live, at least to some extent, normal life, very often they choose this way because that's something that they understand, that they know how it will look like. The other perspective that comes into the scene is that when we think about US and similar markets where we have this insurance and insurance base for your treatment, then depending on how good your insurance is, you can expect that, okay, they can pay maybe a couple $1 million to treat the patient, no matter who the patient is. Is it a baby, a child, or a really senior person? Whereas when you come to many other countries, and especially Europe, and the government pays for that from all the taxpayers' money, and then they compare, okay, we have this cure, probably a cure, for $2 million, or we can, treat like 2,000 patients instead. Well, then the decision is pretty obvious. So with CRISPR, with gene editing based treatment, now what we talk among ourselves is that if we talk about, let's say, younger population, so that's definitely where, you want to cure the child, that it would have this long life and create all the added value it can throughout the life. Or when it's like you live or die, so then it's easier to make a decision that, okay, let's try this one time treatment and hope person will be cured, because there is no other way how to do it. But if we have pretty well established treatment methods which are of let's say competitive price, and it's not life or death decision, then it's probably not the indication you should be going after, at least for now.
Yeah, just to chime in, and just as a disclosure, I'm not a commercial pricing person, but I've been in the industry for a long time, particularly in this space, trying to really address this. And I think it also helps go after that indication where there's a significant unmet need and you're really addressing that. And then from a pricing perspective, one way that is looking there is trying to evaluate what's the total burden to the whole healthcare system over time. So is it 5 years? Is it 10 years? And you take that whole total burden and then determine, is that the price that you can price even as a single administration? So I think that's one thing. And I think that's how most of the pricing has been done. The challenge comes when, from an insurance company perspective, is you're with a private insurer, you have this treatment while you're under their care. That insurance company pays it all, and then you leave to go to the care of someone else. And that new insurance company has a kind of a cured patient, but didn't have to pay all that money out. And I think that, plus how long does that therapy last, is a key learning, because we've seen from some of these really expensive therapies, you dose, it works great for 2 years, 3 years, 4 years, and then it starts to wane. And I think that's a key learning that a lot of us that are in the new therapies now are trying to address. Is there a way that you can have the ability to dose again as you go over time, which then puts it more into a traditional reimbursement model, because it's not a one and done. So it's a complicated question about that pricing aspect, particularly for the one and done. But I think there's been a lot of work in trying to develop that through like Zolgensma, Luxturna, and even Bluebird was kind of at the forefront of that, because they tried to do some aspects around pay as you go. So you get the money up front, but if you start to wane, then you would reimburse and give money back over time from the company.
Great, thank you. We also, before we wrapping up here, would like to open the floor for the audience for any burning questions before finishing here.
We have one here. Niels from HHC Medical. Oh, that's loud. Really interesting topic, the last one. I really hope we get to that one, the pricing one, because I think one of the biggest hurdles to actually commercialize. And I'm from Copenhagen, so I was recently in a meeting with the CEO of Novo Nordisk, and they are not in business of curing anybody, we know that, but he talked about the future where the cure is the answer. And he said that we need to invent a whole new pricing model, the Bluebird, but reverse, so you pay, I call it like a quality life adjusted years as a service kind of thing, right? So do you see that pricing model as realistic? Because I don't see it out there right now. And I think this is one of the biggest hurdles to actually commercialize CRISPR cures for things, for example.
I might say related, right? If manufacturing costs for a lot of things don't get, if they don't come down to a point that we can handle, it's hard to know what the margins are going to be at that kind of scale until we kind of get there. And I think a lot of people have kind of been holding their breath a little bit. I think to the point earlier, is Caszyme like a watershed moment? Certainly. Has it kind of really opened the floodgates the way you might? There's still so many things that we're working out. I think manufacturing and margin's a big part of it. It's wild when you have some of these therapies where the price tag is massive, and you have a company saying, we're losing money making it. I mean, forget all the R&D costs going in, but actually making and delivering that drug at that price, we can't do. It's wild.
But at least what we, to some extent, start to hear, for example, in Europe, and let's say in other smaller countries or regions, is that if at certain point you can actually do that manufacturing locally or regionally, which means still the money circulate in the same country, market, or whatever. So even, let's say, if you have the government paying for that, but that circulates within the country, you actually get the benefit from the taxes. And so I mean, there are ways how to do that. But of course, all these kind of changes, they take time, but I think at least we will definitely see that changing and finding the way how to make it work.
Yeah, and I was going to comment that Blueberry is not the only example, and we have seen a fixed fee and then a success fee, depending on the outcome of this single treatment, and maybe not a lifelong financial model, but we see per year of disease-free, maybe in a short period of time for now. But I think that that's a trend that we will definitely see.
Right. Maybe last question from the audience.
Great comments, Christian. I have an immuno-oncology company for animals, started some years ago. I have a philosophical question. It's very difficult to trick nature, right? And a lot of this discussion here, you got a lot of money, a lot of euphoria in the beginning, which is good. And you went very fast. You had to go fast. You mentioned that all. It's very similar to the early years of transplantation, right? We were happy the heart was beating, and then we learned there's something like rejection. Nature's hard to trick. And then over 2-3 decades, we only found a stable combination of scientific and biological approaches to balance down the immune system, and it's a steady state. You can have a heart, a liver, and you live another 25 years, yeah? And you die of the tram in Zurich downtown, and happily. So here is the philosophical question. If you had more time, and you are the thinkers, the top shots in that gene editing sector, if you could dream 15 years down, what are the scientific, biological elements you probably could imagine when you dream in the night that come together to give a stable, steady state of immunological management, immunological response, nature trying not to be tricked, but being peacefully manipulated with the gene editing you do, and letting that human being happily live and get run over by a tram downtown Zurich in 45 years from now? What would be your thoughts? So
we're finishing on an easy question.
I can take the first part, and I'm not an immunologist, so I won't answer your question, but I'm a big believer of technology and what human beings can do, and we are not tricking nature, and nature will do it better. We are using nature, and we didn't talk about artificial intelligence, but I think this is a huge revolution that we can now talk the protein language. Maybe not as good as nature, but we are getting close there. So I think we will get there in terms of using nature and doing the tools as nature will do. But then for immunology, I'm not an expert. So
yeah, maybe to also comment, and we actually have an expert here on the table, which company is exactly doing that, using the intrinsic function of nature to actually rewrite the genome, which is Paule from AIRNA, which is basically using the intrinsic function of ADA and intracellular protein, which already exist, and just providing the message for actually triggering the correction to happen. And I think this is a good example how we can learn from nature to do those things which normally are driven for viral response mechanisms, but you just recode it to still do what it's supposed to do in nature.
Maybe very quick. So what I'm a big fan is actually to introduce beneficial variants, protective variants that you found in people, basically, because nowadays you get more and more access to biobanks, UK Biobank, all of us, and then you can find actually variants in people which protects them from certain diseases. And this is something, and this is not something which is happening in 15 years. This is actually what we are already doing, is to introduce those protective variants which helps people to lower their LDL, but also help them against neurodegenerative diseases, basically.
Great. With that, I thank all my panelists here for a fantastic session, the audience for your patient and great questions. Thanks, everyone.